Hi everyone, my name is Brooke Sasha. I'm a recent graduate of Cal Poly San Luis Obispo, and I'm a current grad student at the University of Wisconsin-Madison. And my project is titled High Definition Transcranial Direct Current Stimulation Enhances Figure Ground Perception for Part-rearranged Images. And I completed this project in conjunction with Laura Cashamani at Cal Poly. So humans most often rely on their sense of vision to perceive the world around them. Perceiving even a single object requires multiple brain areas, and it's often accomplished through a process called figure ground perception, in which we break parts of a visual scene down into the figure and the ground. The figure being the part of the visual scene that stands out and has a definite shape, and the ground being appearing to be located behind the figure. So in this example picture here, the corgi is the figure, and the ground is the tall grass located behind the corgi. One really great example of figure ground perception is this famous face-space illusion, in which the figure and ground are ambiguous and can actually change depending on your perception. So right now you might see the figure as the black face in the middle, but you might also see the figure as two faces and white. And it's important to note that you can't actually appear or view both regions as figure at once. You have to break it down into the figure and the ground. And again, your perception can change and there's no right answer. So the brain area that's involved in this process is called the Lateral Occipital Cortex, or LOC for short. And the LOC plays a key role in object recognition, and it's active when looking at objects that have a defined shape. And it doesn't matter whether they're familiar or unfamiliar, or two-dimensional or three-dimensional. Additionally, the LOC is not active when looking at something like a texture or visual noise. So what we wanted to know is, how is the LOC and figure ground perception affected by brain stimulation? So we used a brain stimulation technique called transcranial direct current stimulation, or TDCS, in which two large electrode pads are placed on either side of the head, like in the picture on the slide, and a mild electrical current is applied. Essentially, it makes neurons either more or less likely to fire, so it's not directly altering the neuronal firing rate, and effects last for up to 90 minutes. Additionally, there's only one or two studies using TDCS to look at object perception, and one study actually found enhanced perception of objects and faces. However, there is currently no research using HD-tDCS, which is high definition TDCS, and a newer form of the conventional version of TDCS. Essentially, the difference is instead of two large electrode pads on either side of the head, there's 5 smaller electrodes arranged in a ring configuration. So you can see in the image on the slide, there's 5 electrodes in the ring configuration, and because of this, the focality of the stimulation is actually improved, meaning that we're actually more likely to stimulate the area that we want to stimulate due to the use of 5 smaller electrodes as opposed to 2 larger ones. Additionally, since you're stimulating a less overall area of the brain, it's actually safer than conventional TDCS as well. So in order to target the LOC, well, first, we chose the right LOC based on literature showing that compared to the left LOC, the right LOC was more active when viewing objects. There wasn't a ton of research on this, but based on prior literature, we decided that the right LOC was the best decision. In order to make sure we were actually stimulating the area where we wanted to stimulate, we used a modeling software that gave us our expected current flow. So in panel B, you can see that the areas in red of the brain were the areas receiving the highest amount of stimulation. So that's directly corresponding with the right LOC. And in panel A, you can see the 1020 EEG system layout with the electrode placement that corresponded to stimulation of the right LOC. So the central electrode is placed at PO8 in red on that image, and the 4 surrounding electrodes are denoted in the turquoise light blue color. So this brings us to our research question, and that is, can figure ground perception be influenced by brain stimulation? And we hypothesized that stimulating the right LOC via HD-tDCS will enhance figure ground perception. And a note is that there are no studies using HD-tDCS to assess figure ground perception to date. And this is really important because figure ground perception is how we perceive objects. So here's a quick overview of our methods before I get into more specific details. First, participants were administered a pre-screening, both online prior to the experiment, as well as in person in the lab when they came in for the study. This was to ensure that it was going to be safe for them to participate. Next, they received 20 minutes of offline stimulation or sham, with offline meaning that the entire duration of stimulation was completed before moving on to the behavioral task, and sham being the placebo or control condition in which participants believed they were going to receive stimulation when in fact no currents were administered. Third, they completed the behavioral task, which I'll talk about next. And finally, they completed a post-experiment questionnaire so we could learn more about their experience in the study. So a little bit about our behavioral task. So participants viewed sets of stimuli in which there was a half black region and half white region separated by a vertical border down the middle. Half of these stimuli contained a familiar object termed familiar stimuli, and the other half contained the same familiar object except the parts were rearranged to appear unfamiliar, and we call these the part-rearranged stimuli. So if you look at the example familiar stimulus, which is on the left, you can see that in white there's a seahorse. That would be a familiar object. And then in the part-rearranged stimulus, the same seahorse is in black, except it doesn't quite look like a seahorse anymore because the parts have been rearranged. So participants were asked to choose which side of these images looked more like the figure to them. And we hypothesized that those who received stimulation would choose the side with the familiar or part-rearranged region more often than participants who did not receive stimulation. So here is what each trial looked like. I began with a fixation cross in the middle of the screen, and participants pressed spacebar to begin the trial. Next, the stimulus flashed for only 50 milliseconds, which is a very short period of time. You'll see in just a second. And that was immediately followed by a 200 millisecond mask. Finally, there was a blank screen until they responded. I'm so sorry about the sirens. And the purpose for the mask is because we wanted participants to process these stimuli unconsciously. In other words, we didn't want participants to rely on prior knowledge in order to process the stimuli. So next, I'm going to show you a example trial. Just be sure not to move your eyes from the black fixation cross because it goes very quickly. 3, 2, 1. Okay, so if you didn't catch it, it's okay. That's very common, and pretty much all the participants felt the same way. So what you actually saw was an image, I think it was of a toilet. Yeah, and if you did see it, that's very impressive. So moving on to data analysis and our results. So we completed a 2x2 mixed design ANOVA in order to determine whether the critical region was chosen more often based on condition, stimulation or sham, and stimulus type, familiar and part rearranged. So the outcome measure in all analyses was the percent critical region chosen as figure. And the critical region is the side of the stimulus containing either the familiar or part rearranged region. So in the familiar stimulus, you can see that the critical region is a snowman, which is in white. And then in the part rearranged stimulus, the critical region is the part rearranged snowman in black. So here's what we found. Most interestingly, there was a significant interaction such that stimulation did enhance perception, but only for the part rearranged images. In other words, stimulation had no effect on participants' perception of familiar stimuli. This is really interesting because it is only partially in line with our hypothesis, as we thought it would enhance both of them. So that's really interesting. The second thing that we found was a significant main effect within the sham condition, such that participants in the sham condition chose the critical region more often for familiar stimuli than the part-rearranged stimuli. So in other words, they were more likely to choose the critical region when it depicted a familiar object. And this is actually replicating previous research using these studies, using these stimuli. So that's great that our results were in line with previous literature there. So just to quickly recap, the purpose of this study was to investigate the effects of HD-tDCS on figure ground perception. And our results were partially in line with hypotheses, such that participants in the stimulation condition did have improved perception, but only for the part-rearranged stimuli and not the familiar stimuli. We also replicated previous literature such that in the sham condition, participants were more likely to choose the critical region when it depicted a familiar object. So the reason we think maybe that the stimulation had this interesting effect on only part-rearranged stimuli is that it may have helped participants see familiarity in somewhat ambiguous stimuli. In other words, stimulation may have taken away some of the ambiguity in the somewhat unfamiliar objects. So again, this was the very first study to stimulate the LOC using HD-tDCS and to look at figure ground perception. Figure ground perception is an almost instant process, and we use it all the time in our lives for those of us who are sighted. However, for individuals with visual impairments, this process may be slower or less efficient, and this study may lay the framework for more future clinically oriented work. For example, an individual with brain damage due to a stroke or a traumatic brain injury. So the next step might be to implement HD-tDCS for the clinical population. But to conclude, object perception is extremely important and we use it every day. So having a decreased ability can be very impairing, and HD-tDCS has a lot of promising implications, and more research is definitely needed to see if these effects carry out throughout other situations. And I would also like to acknowledge Dr. Laura Kashamani for assisting me with this project and being a wonderful advisor. And I would also like to thank Bailey Buchanan, Chris Oseguera, June Pauley, Gabe Lavezzi, and Calvin Tower for their assistance with data collection. Here are my references. And if anyone has any questions, feel free to leave a chat message and I will answer those. Or you can also feel free to email me. My email is sasha@wisc.edu. It's also on the slide. Again, thank you so much for listening to my presentation. I really appreciate it, and I hope you all have a wonderful time at the rest of the conference. Thank you.